Okay. All right. Well, thank you everyone for coming. And this is our first tree steward training of 2022. And we are really happy to start it with Jim McGlone, who is going to be presenting all about tree physiology and how this ties in with pruning, which you're going to get the practice on over the next couple of weeks. And I never know how to introduce Jim, except that he's been such a stalwart of our tree steward training. And those of you that were on the tree walk with him on Saturday know that he is such a fountain of information, and it's so wonderful to have him. So without further ado, Jim, I'm going to turn it over to you, and you tell me if I need to do anything on my end.
I don't see anything that you need to do on your end. I'm going to start sharing my screen, and we'll get started. As Nora said, my name is Jim McGlone. I am an urban forest conservationist with the Virginia Department of Forestry. And tonight we are going to talk about tree physiology and applications to pruning. Before we get started, I do want to point out that although we're going to be using the word tree tonight, I am referring to all woody plants that are either angiosperm dicots or conifers or ginkgos. An angiosperm dicot is sometimes referred to as a hardwood or our local deciduous trees. The reason I'm excluding angiosperm monocots, which would be palms and bamboo, is that they behave very differently from the other woody plants. So we're going to start talking about tree structure. Now, this is what trees aspire to. This is from a program called the Landscape Below Ground at Morton Arboretum. And as you can see, the tree canopy and the root system, at least of the trees we're talking about tonight, are dendritic. And that means that they branch. They go from big pieces to little pieces, or little pieces merging into and eventually forming a big piece we call the trunk of the tree. One of the things to notice in this diagram is that the canopy and the root system have very different geometries. And one way that you can think about this is if you were making a pizza from scratch, you would start off with a ball of dough, which would be the canopy, and you would flatten it out into a disc that would be the crust of the pizza. And for the most part, the mass of the tree canopy and the mass of the root system are roughly equal. Now, there are going to be lots of things that can cause that to go one way or the other. In particular, wind, which can put stress on the root system, can cause it to grow bigger to help hold the tree in place. But they are both dendritic, and they both start from what is sometimes called the root collar, or the base of the trunk, or the trunk flare, or various different things. But it all means that spot where the tree stops being root and becomes trunk. So the systems that we have in the canopy and in the roots, as I said, are similar, but they're not the same. And what we see is there are certain structures that correlate to each other. In the canopy, we can start with the leaves, which are small structures that support the energy system. This is where photosynthesis takes place, and leaves are temporary collecting structures. They collect light and carbon dioxide to support photosynthesis, but they are lost, whether it's an evergreen or what we would more commonly call a deciduous tree. they do lose those leaves at some point in their life. In the root system, we call these absorbing roots. These are very small structures that are, again, temporary, and they support the energy system through the collection of water. Because water and light and carbon dioxide are what we need for photosynthesis. They also collect nutrients, which supports the tree in a different way. That's what the tree uses the energy on. And those absorbing roots are not what you normally see when you dig in the ground. The absorbing roots are attached to them. As I said, they're very small structures, only a few cells wide, and the tree is constantly growing and shedding those. In fact, the amount of biomass that's grown and shed as these absorbing roots is equal to or greater than the amount of leaf mass that is shed every year. Supporting these small absorbing structures are either the branches and twigs in the canopy or conductive roots in the root system. And these are those small roots you might see while digging up through about 2 inches in diameter. And then supporting the entire crown are the scaffold branches. These are the really large branches that are connected directly to the trunk. They are a weight-bearing structure in the tree. The buttress roots are also very large roots that are right at the base of the tree, and their primary function is to support the tree and hold up the weight of the tree. So as I said, canopy and root systems are similar, but they're not identical. And the biggest difference is the geometry of the crown versus the geometry of the root system. And also, for what we'll talk about later on, keep in mind the root system is not as subject to gravity because the soil is supporting the root system as the canopy is, because gravity is pulling down on all the branches and everything. In a tree, we have 3 types of tissues. There's meristem. These are the cells, the only cells that are capable of cell division, that is making new cells. And they're also capable of differentiation. So this is where growth occurs, is in the meristems of the tree. We have vascular tissue, which would be the vessels in the dicots or tracheids in the conifers. They are what conduct nutrients and water from the roots up to the crown, and phloem, which translocates photosynthate. The phloem is just inside the bark and it's moving sugar around, either from the leaves into the other living cells in the tree or into storage, or at this time of year, it's going from storage as starches, it's being turned back into sugars and being pumped up into the canopy to support flowering and leaf production and shoot production. Everything else that's not meristem or vascular tissue is called ground tissue. This would be the fibers in the stems and the seeds. These are almost all dead cells, so they form and they die almost immediately, and then all of the stuff on the inside of the cell that kept it alive is sucked back into the plant to be reused. The epidermal cells, or the epidermis, is the outermost skin. It's not just bark. The root system does not have bark. It's got epidermis. And the function of the primary function of the epidermis is to control gas and water exchange and to keep water in the tree. Therefore, epidermal cells, particularly the bark of the tree, is what we call hydrophobic. That is, it repels water, which is why bark makes really poor mulch. So if you are going to mulch, don't use bark. In addition to repelling water, it will also float away and make a mess outside of wherever you're mulching. The last type of ground tissue is the parenchyma. And the parenchyma is very important. It does lots of things. The main things that it does for the tree is this is where photosynthesis takes place. This is where carb storage, that is energy in the form of starches, takes place. And the parenchyma cells are the ones that produce all of the toxins that defend the tree. So for example, in an oak tree, it is in the parenchyma where tannin is produced. The parenchymal cells are living but non-reproductive cells. That is, they have all the machinery, but they cannot make new cells. The only tissues that make new cells are the meristem. So if we look at this micrograph of a water oak, Quercus nigra, we can see these different types of tissue and parts of the tree. On the outside over here, this is the bark. And you can see some of the cells in here. Because this is an oak, it actually does have a third type of meristem that makes those cork cells. Most trees don't have that. Just inside the bark is what's called the phloem. That's this tissue right along the outer edge of this dark line. And that line is where the cambium was when this tree was alive. It's obviously, since I'm taking pictures of it with microscope, it is dead, but that's where the cambium is, and we'll talk more about that. That's one of the meristems in the tree. Inside the cambium, you have the xylem, or the wood of the tree. This is where water and nutrients are conducted. Sugars are conducted out here. The fact that that cambium is right below the bark is why you don't want to ruin the bark, because you can, if you wound or disrupt that cambium, you can kill it, and you make it hard for that to make new wood and new tissues. In the xylem, we have vessels. That's these very large pits, but there are also small pits through these kind of cream-colored lines. There are fibers. That's the brown stuff here. Those are there primarily to support the tree. Remember, the fibers are dead tissues. The vessels are dead tissues. The phloem does have live tissue in it. And then the cream colored part surrounding the vessels and also snaking through the fibers here is parenchyma. And all trees have these rays of parenchyma that you can see here that extend out to and through the cambium and connect to the phloem. That's how the sugar gets from the phloem into the wood, where it can be stored or used for growth or used for life functions. So those are the parts of the tree. We'll talk more about the parenchyma, especially when we get to talking about how the tree controls decay. Are there any questions so far before we move on and start talking about how trees live? Okay.
Yeah, wait, I have a question. I'm still getting that double feedback, and it's very hard to follow the words, especially the scientific ones.
All right.
So Martha, I think you're on twice. At least you were on before twice. Can you, let me look.
Should I sign out and sign back in?
Well, the last time I checked, you were on twice. Let me look and see.
Well, if don't, I'll just roll with it and get the info sorted out from my notes.
You're somehow in the system 2 times, which is why you're getting the feedback. I'm not sure how you're doing that. Are you on your phone and your computer?
Well, I'm on my computer, but please don't interrupt. We need to get on and all this important stuff.
Well, that's why you're getting it twice. I think everybody else is fine. Okay. Sorry about that. I'm not sure why you're on twice, but you are. You're in here twice. I see you in here twice. All right.
Okay.
Anyway, go ahead, Jim.
All right. Well, let's move on then to tree physiology. And this is going to be very, very basic. See, there we go. So the very basic outline of what I think of as tree physiology is essentially how the energy system works to produce the tree. Plants create biochemical energy in the form of sugars by harnessing the energy in photons. That sugar and then the water and nutrients and oxygen are moved around through the plant through the vascular system. Some of those sugars wind up in the living cells where they get burned in the presence of oxygen. This is the same thing that happens inside your body. And that cellular respiration uses electrons to create adenotriphosphate molecules by adding a phosphate group to an adenodiphosphate molecule. And that's an energetic molecule that then can go off to the ribosomes where it can fuel the production of new molecules as directed by the DNA of the plant. And those molecules are used either to repair the cells, to produce those toxins, those secondary chemicals to defend the tree, or if it happens to be meristem, to promote cell division and to make new tissues. And so the photosynthesis takes place in the parenchyma, and cellular respiration takes place mainly in the parenchyma and the meristem. As I mentioned, there are some live tissues in the epidermis as well as the phloem, and that you would get cellular respiration taking place there. But the primary stuff, the important stuff, is happening in the parenchyma and the meristem. So this is what photosynthesis looks like. This is a cross section of the leaf. You can see these cells out here that are called the cuticle, and the epidermis are what keep the leaf together. Their function is to try and keep all the water and moisture and everything inside the leaf. Here is where you definitely have living cells, or these guard cells. They act like one of those coin purses that you squeeze to open, and that opens the stoma, which allows carbon dioxide to go in and oxygen and water vapor to leave, to go out through the leaf. And the basic equation for photosynthesis is 6 carbon dioxide and 6 water molecules. And the leaf uses photonic energy to break these molecules apart, rearrange the atoms, and produce a sugar molecule and 6 oxygen molecules. If you look at this really quick, you'll notice there are the same number of carbon, oxygen, and hydrogen molecules on both sides of the equation. So it is not correct to say that the tree is creating oxygen. It is correct to say the tree is releasing molecular oxygen. And as I pointed out here, the CO2 comes from the air through the stomata, and the water comes from those absorbing roots in the root system and up through the xylem into the leaves. And the output is sugar plus this waste product that we call oxygen. Cellular respiration is kind of the reverse of that. So the sugars find their way into a living cell, into the mitochondria. That's this little structure here. And in the mitochondria, that sugar is burned in the presence of oxygen to produce 6 CO2 and 6 H2O. This is why it's kind of the reverse of photosynthesis, because the byproducts of cellular respiration are the same as the inputs to photosynthesis. And then instead of photonic energy, you have this energetic molecule. So in a sense, if you want to get into the quantum dynamics of it, the plant through photosynthesis and cellular respiration is taking photonic energy and turning it into energized electrons that then are used in the ribosomes, which you can't really see, their little dots spread throughout here, that are making new tissues or the toxins or the cell maintenance using that the energy in the ATP and the nutrients that come up from the root system. And this leads to a model of tree energy. So down here you have the roots. It's pumping water up into the leaves where it's combining with carbon dioxide and sunlight to make sugars, which are going into either storage or into the living cells. The living cells are receiving nutrients and oxygen from the root system, and then they use cellular respiration and their machinery to make these new tissues or maintain the cells or produce those toxins. This model will become useful later on when I come back and talk about tree diagnostics in a little over a month. There's one other important thing that the tree does with its sugars, and that is create exudate. Exudate is mostly sugar, but it is, there are other chemicals in it, and the purpose of exudate is to attract beneficial microbes. Now, the microbes may be beneficial in the sense that they are laying down an additional layer over the epidermis that is protecting the tree from pathogens simply by being an additional layer that the pathogens have to get through. But they're also maybe actively protecting the tree by killing the pathogens. There's a lot of microbial warfare going on in the soil, and we have harnessed that for almost all of our antibiotics, whether they are whether it's from bacteria or soil fungi. So the tree is also trying to harness that with these exudates. One of the other things that is attracted are certain types of fungi called mycorrhizal fungi, and they form mycorrhizae, which is fungus root, is the translation of mycorrhizae. This is a symbiotic structure that's created on the root system. In the picture, all those white fluffy things are mycorrhizae. That's actually a goldenrod root. And what the fungus is doing is it's collecting water and nutrients. It's essentially extending the root system of the tree. But because it's fungus, it's not a temporary thing. They are moving around. They're extending that root system, but they are also potentially creating a connection with other trees and plants. And so water and nutrients and other chemicals can be exchanged through that mycorrhizal fund network. What the fungus is getting out of it is sugar. Very few of these fungi are specialized. As far as I know, there's actually only one group that specializes. These are the Iroquois mycorrhizal fungi, and they specialize on the family Ericaceae, or the heaths. Now that might sound impressive until you realize that there are 30 or 40 genera in the family Ericaceae, one of which is rhododendron, which has over 1,000 species. So even though they are quote unquote specialized, it's not that great deal of specialization. The other thing to understand about these mycorrhizae is you can go to the nursery and you can buy spores. I advise against it simply because one branch of research that looked into it found that about 75% of the spores that you buy are dead. So that's not going to help. The other thing is, although there is good scientific research that this stuff helps in a laboratory setting, there is no scientific research that suggests that it helps when you go out into the field. And that's because, as I said, these things mostly are non-specific and they're everywhere. So buying that packet of mycorrhizae and adding it to your planting, it won't hurt anything, but it's sort of like throwing a bucket of water in the ocean. It just doesn't have much effect. So that's the basic physiology and the sugar production and usage. Any questions?
So far, no questions.
Okay. Let's go on and talk about how trees grow. Now, remember I said that the only tissue that can support growth is the meristem. And in trees, there are two types of meristem. And this is where trees are different from other herbaceous plants and from the monocots. So all plants have a primary meristem. These are also called buds. And these are things that can these are cell tissues that can elongate into shoots or roots. Along the way, it produces a cortex, which is the nascent vascular system. The epidermis, it'll produce lateral buds in some cases. It can become a leaf, it can become a flower. All plants have this. There are different places in plants. Grass, for example, has primary meristem that's located in the root crown at the base of the plant. That's why you can run a lawnmower over it and not hurt the plant, and it just grows back because it's growing from the bottom up. In trees, the growing meristem, primary meristem, is all located in buds at the tips of branches or roots. There is suppressed meristem in the dicots along the stems, along the branches, along the roots. In conifers, you don't have those suppressed buds. So pruning conifers becomes different from pruning dicots, because when you prune a dicot tree, if you do it wrong, you're going, or if you do it, I won't say wrong, but if you do it in a certain way, you are going to promote the release of those lateral buds that will make new shoots below the cut. In conifers, that usually doesn't happen because they have few, if any, suppressed lateral buds. Now, where the woody plants, the woody dicots and the conifers and the ginkgos differ from the herbaceous plants and the monocots is they have secondary cambium or secondary meristem, which is called the cambium. This is the tissue that's located between the bark and the wood that I pointed out on the micrograph. And this is what produces the xylem, which is the wood on the inside where water and nutrients are moved, and the phloem, which can become part of the bark or it can be reabsorbed, and that's the tissue that moves sugar around. And remember that part of the xylem is also that the stem parenchyma and the fibers that you find in the wood. Palms don't have this, so they do not get bigger around as they grow. They have one meristem bud at the top of the plant. They have another one at the base of the trunk, which makes the roots. Palms also don't have a dendritic root system. They have a fibrous root system, so their roots don't branch. That's why I said we're not going to talk too much about them tonight. So here is my attempt at showing you graphically what's going to be happening in a few weeks. Out there right now, there are woody plants that have shoots on them. The shoots are would be this blue triangle with a terminal bud, also called an apical bud. Right now, those buds are waking up. They're producing chemicals. They may also be producing new cells. That's why they're getting bigger. And eventually what's going to happen is in a couple of months, they're going to start elongating and making new shoots. Along the way, they may produce lateral buds, they may produce leaves, they may produce flowers, depending on the structure. Some of them, the terminal bud, especially on, for example, azaleas, that big fat terminal bud is actually a flower bud. The shoot buds are tucked in underneath, and so there will be new shoots coming off under those flowers. But after the primary meristem becomes active and makes that shoot, the secondary meristem becomes active and makes a new layer of wood over last year's wood. So you might go out there in, say, late April or May and see something like this. This is a Viburnum Dentatum. If you look in the upper left, you can see my fingers holding the brown stem of the tree, pulling it, or the shrub, pulling it down so I can get a picture of the new shoot. So all of that green tissue is the new shoot that grew from where my fingers are down all the way off the picture. And as you can see along the way, it made new leaves. And at the end of that shoot, there will be a bud called the terminal bud. And in this case, and frequently, there will also be what are called lateral buds or axillary buds. And if you look really carefully up here, you can see there are two little shoots going off to the side. So those are smaller buds that were tucked in under the terminal bud that led to the new growth that happens in the spring. So trees and shrubs elongate from their tips. So if you nail something to the side of the tree or you carve your initials in the tree, it will never get any higher off the ground as the tree grows. It's only, the trees are only growing from their tips. Unlike, as I said, a lot of herbaceous plants, they have that primary meristem in the base of the plant and they grow their shoots up from the base. Another thing that we see happening in trees that doesn't happen in herbaceous plants or the palms or bamboo is lateral growth or diameter growth. So they get bigger around every year. And the way this works is pretty straightforward. This is a piece of wood from last, or a shoot from last year. This is living xylem tissue. It has living parenchymal cells in it. It's part of the sapwood of the tree. So this is tissue that's conducting water. This year, we'll get a new layer of that living sapwood that's conducting water on top of last year's layer. But last year's layer is still alive. It still has living parenchymal cells in it, and it is still being used by the tree to conduct water. So this is what's causing the tree rings as we do this. Eventually, it will get to a point where that interior sapwood is going to die and become heartwood. And when I say die, keep in mind, this is what we make our 2x4s for. So it's dead, but it's not rotten. It's still structurally sound wood. It's just no longer has any living cells in it, and it no longer is conducting water. And as I said, this is what causes the growth rings. So going back to the micrograph of the Quercus nigra, you can see I've kind of drawn these two red lines. It's actually 1 year's growth. Quercus nigra, all the oaks are what we call ring porous. So you can see they produce these great big vessels early on in the growing season, and then they produce smaller vessels later in the season. This creates two types of growth every year. The first growth is called early wood, or sometimes it's called spring wood. The later growth is called late wood or summer wood. All trees exhibit this pattern where they grow for a while and then they stop. And at the end of the growing season, just inside these heavier, this line of large vessels is a specialized tissue that's put down at the end of the growing season by the cambium. This is a very dense, tough tissue, and the tree puts that in place to try and keep decay from penetrating further into the tree. So as we look at this, the space between the two red lines is a single year's growth, and then the blue line separates summer or early wood from late wood. It's not that easy to see that separation. Sometimes it's not even easy to see that growth, annual growth in trees. This is sugar maple, Acer Saccharum, and this is what we call ring diffuse. That is, it has produces vessel elements of approximately the same size throughout the growing season. But because of that, that distinction between early wood and late wood is very hard to see if it even exists. It's also really hard to see the growth rings, although they are still there, because even these ring diffuse trees are producing that decay barrier at the end of the growing season. So if you look at this picture, you might think those bright lines that are going from the upper left to the lower right are the growth rings. Those are actually the parenchymal rings, parenchymal rays. The growth rings are here, here, and here. Like I said, even under magnification, it can sometimes be hard to see. So when you're looking at this, just looking at a cut piece of wood, it can be really hard to see those growth rings. But they are there, and they are delineated by those barriers the tree is putting down at the end of the growth, at the end of the growing season. I mentioned the heartwood. The heartwood, as I said, is dead tissue. This is being actively created by the tree. There was a time we thought that the tree just stopped using it for moving air, water and nutrients, and dumped a bunch of garbage in there. But now we know this is very carefully created by the parenchymal cells as they die. They're infusing that wood with defensive chemicals. They may be changing some of the structure of the wood. Particularly the white oaks get these things called tyloses. These are essentially little airbags that the parenchyma blows out into the vessels, and they're there to try and prevent vertical movement or movement of decay along the heartwood in the tree or in the branch if it's a big branch. And usually the heartwood is darker than the sapwood. So if you look at this cross section here, you can see the heartwood in through here is this dark brown wood, and this was the living sapwood when the tree was cut down, this lighter color. Also notice there was a branch here, and see how the branch is had heartwood and is connected to the heartwood in the center of the tree. So with all of this, we can look at a cross section of the tree. You have that dead heartwood in the middle that relies on the chemical and in some cases physical transformation of the xylem tissue by the parenchyma as the heartwood is created in order to protect it from decay. The sapwood is the living wood, and it is relying primarily on the parenchyma to protect it from decay. There are no secondary chemicals being produced and infused into this wood unless the tree has been wounded. Then you have that very thin layer of the cambium, the phloem, and the outer bark. In the picture on the right, this is a white oak stump, Quercus alba. I took this picture about 6 months after the tree had been cut down. The lighter colored wood, the lighter colored tissue over here is the heartwood. Notice that it's getting a little staining as fungi try to get in there and decay it. This dark band is the sapwood. Now, I said earlier that the heartwood is usually darker than the sapwood, and when this tree was alive or had just been cut down, that was the case. But remember, the sapwood relies on active defenses from living tissue, and after the tree was cut down, all that living tissue that normally protects the sapwood died. So it's being heavily invaded by fungi that are decaying it, and that's why it's black. It's been stained by those decay fungi. And then out here you would have the cambium and the phloem and the bark, but you can't really see that in this picture. Now I want to talk about one type of specialized growth, and then we'll stop for questions. and that is reaction wood. Reaction wood is produced by the cambium. So this is again secondary or meristem that's producing these tissues. And there are two types of reaction wood. One is called wound wood, and this is created by the callous tissue in the cambium. You may hear somebody refer to these lips that you see around a burning wound or around a wound as callous tissue. That's not the callous tissue. That's the wound wood. The callous tissue is microscopic, and it is essentially when you wound the tree, you create a hole in the cambium. And so these are the cells the cambium produces at the edge of that wound. And as the tree as it grows more and more wound wood, that hole in the cambium is going to get smaller and smaller, and eventually that callous tissue will go away as the tree finally seals over the wound. There is also reaction wood that's created by stress on the cambium. And that stress can be caused by a variable load that would be swaying, where the cambium is going into compression and tension in a repeated cycle as the tree sways in the breeze. But it's also created by a static load, which would be gravity pulling down on the weight or gravity pulling down on the tree. And so in branches, we talked about a little bit about this on Saturday. The top of the branch is in tension, the bottom of the branch is in compression, and that continuous static load causes the cambium to grow wood in different ways to combat that static load. But if there is decay in the tree and it creates a cavity, that can also shift that static load of the weight, particularly in the trunk, the weight of the tree around that cavity, and can cause the cambium to grow new wood in response to those cavities and those shifting loads. So this is reaction wood from a static load. So in this tree, you can see it's got this kink here, and right in here, there is this nice ridge of wood that's been formed because of the static load on the cambium down here. And this is what it would look like in cross section, where here's your center of growth, and because of a static load that caused the cambium to make more wood on this side of the tree than on this side of the tree. So frequently that's what your branch cross sections look like. You can also see static loads in the tree, like I said, particularly in the trunk, and this can be important when you're looking at the tree and trying to figure out or identify problems or weaknesses in the tree. Here on the left, this is called bottle butt, and it's called bottle butt, and it's caused because there is decay in the middle or in the heart or in the butt of this tree down in here, and that is causing new loads on the cambium that cause it to create this reaction wood here. In this tree, it's a little more subtle, but you notice there is a bulge right in here, and that's because there is a cavity, and you can actually see where the tree has closed over that cavity. But as it responded to that change in loads at this point, it built reaction wood to compensate for the loss of some of the supporting tissue there. When we're looking at variable loads, this is what causes the taper in the base of the tree. You can really see the taper on the right-hand side, the part that's kind of in shadow here. You can see that is from the tree swaying in the breeze. The other thing that responds to reaction wood or to this variable load is the roots that you can't see in the base of at the base of the tree. Those buttress roots get much bigger around. Remember, I said the conductive roots are up to about 2 inches in diameter. You will find a thing called a root plate around the base of the tree, where root diameter suddenly drops down to about 2 inches in diameter. So this big root is going to grow out about 10 feet from that tree and then suddenly become a 2-inch diameter root. And that's all because of the response of those roots to the forces associated with the tree swaying under that variable load, putting them in tension and then compression and then back into tension. Any questions so far?
Yeah,
No?
there's one. It was from way back when you were talking about, Emily asked, is primary/secondary here what you were speaking about? Is it the same as the apical and the lateral, which was the terminology in the handbook?
Yes, I believe so. I didn't go back and reread the what you call it, the book, but I would assume apical is certainly the primary meristem. The lateral meristem, it's usually called the cambium or the secondary meristem, but it can be the lateral meristem, which is growing the new wood.
Okay, and here's another one. Is there a transition zone between the roots and the trunk?
There is, and it's really important when you are planting trees. That transition zone, in terms of the forces, not so much, but that is where the tree stops growing bark and starts growing root epidermis, and it should be right at grade, at the ground, at the level of the ground. So I can back up. So that transition zone is right here. It's sometimes called the root plate or the trunk flare or the root flare. It goes by a lot of names, but that's where it transitions from being roots, which have a little bit different anatomy and growth patterns, to the trunk and the top of the tree. That's, as I said, one of the big differences is the tree grows bark above ground, but it doesn't grow bark below ground. And that's because the tree doesn't need the bark to protect it from things that you see above ground, in particular solar radiation, when you're talking about roots. Okay.
That's all for now.
Speaking about roots, let's talk a little bit about root growth, because it's a little different than trunk growth, mainly because roots don't really have to contend with gravity, but they do have to contend with soil. So the picture on the right shows the growing part of the root. Again, just as with the others, you have primary meristem in the roots. That's in the meristematic zone. This is what's making new cells. And those cells then elongate based on chemical signals. And that's really important, because as that root elongates, it's got to push against the soil. So one of the things that the primary meristem makes in the roots that it doesn't make in the canopy is this root cap. These are epidermal cells, and the function of the root cap is to protect that meristem as the tree roots elongate, but it also lubricates the root as it pushes through the tree, because they're sloughing off and they're helping ease the passage of the root through the soils. And then after they elongate, they will start differentiating into new things. And the same thing actually happens above ground a lot. So pay attention to how the shoots expand in the spring, and notice that you'll see elongation, and then you'll see little tiny leaves coming off the side, and then they will start to expand more. So you're getting a little bit of that elongation and then differentiation. And as they differentiate, they're going to create root hairs. So this is an absorbing root. Not all roots still have root hairs. Keep in mind, a root hair is a modified epidermal cell. So this picture is greatly magnified. This is measured. This is like microns. The cortex, which will become part of the vascular system, may be producing secondary meristem, because once the root has extended, it will, just like above ground, you get growth rings in the roots and you get new growth in those roots every year. So this is what a root system might look like. This is a red maple, Acer rubra. And what you can really see in the picture are the conductive roots, which are all of these things that reach out. Notice that also the orientation of the roots. It's just like that picture I showed you at the beginning. The absorbing roots would be attached to all of these finer roots down here. And you can start to see the heart root and the beginning of that root plate and those buttress roots. Now, obviously, this is 1-year-old seedling, so buttress roots are not that important. There's not much that strain on them from wind. Another important part of growth is branches. So as I said on Saturday, branches are not attached to trees the same way our limbs are attached to our torso. If you notice in the picture here, one of the important things is that branches go down into the trunk of the tree, all the way down to the pith. So again, you get primary growth, which are new shoots. And then that secondary growth starts at the top and it goes down. And a result of that is the branch is going to have its secondary growth before the tree does, which means there is going to be new xylem tissue from the branch that's going to interconnect with the old branch xylem tissue. And then the trunk is going to grow new xylem, new wood that overlaps that and kind of wraps it. So you get this interweaving and this mesh between the branch tissues and the trunk tissues, and that's particularly true below the branch. And as I said, good branch attachment goes all the way into the center of the tree. It starts when the tree is that size, it starts putting out what will eventually become branches. On the outside, what you will see is the branch angle or the crotch here is open. It's frequently described as U-shape. It can be as much as 90 degrees, which is almost here. And it's hard to see in this picture. I'll show you a better one in a minute. But there is what's called a branch bark ridge, which is where the bark of the tree and the bark of the branch comes together and pushes up the bark out away from the connection. Here is a diagram from Alex Shigo, who is in many ways, he's not the father of arboriculture, but he's the one who put it on more of a sound scientific basis. Throughout the 70s and 80s, he cut up a lot of trees with a chainsaw to figure out all this stuff out. But you can see, you have the branch, it's growing new xylem over the old branch, and that xylem is going down because it needs to connect with the tissues that are bringing water and nutrients up that high. And then there is the trunk tissue wrapping over it to keep it strong. And there's that branch bark ridge. And this is what it looks like on the inside. This is unfortunately my Quercus fellows branch ripped off of it during a storm, and not wanting to waste anything, I took a picture of it. But you can see how the wood is wrapping inside there. And here's what a good branch attachment looks like from the outside of the tree. This is a Cornus Florida on the side of my house. It has this really classic U-shaped connection, and you can see that branch bark ridge right in through here. You'll hear a lot more about branch bark ridges in your pruning classes on these following Saturdays, because they're important for figuring out where to make your cut. Here is another smooth bark tree, but you can see the branch bark ridge. I think this is Acer negundo, the box elder. My wife saw this tree when we were out walking in the woods and thought it was diseased. And I looked at it and said, no, it's supposed to look like that. You can also see the lenticels on here where you have oxygen going into the tree and carbon dioxide coming out. You can also have bad branch attachments. And this is where you usually have a very narrow crotch angle, a V. You don't have a branch bark ridge. This is actually a seam. And as you can see here, and we'll see in the next picture, there is bark that's included in here. So the attachment point of this branch is not here as it would be in a good branch union. It is way down at the bottom of the picture. And what you have in this area are two pieces of trunk or two pieces of tree that get bigger around every year, and they start pushing against each other. That's why you have, if you can see, this is starting to form a raised seam here. And the problem is when they're pushing against each other, like you have here, is that when you have a storm come along, maybe a wet snow like we had at the beginning of January, it creates additional load on this branch and it can rip it down. And you can see the included bark up here and the rip that it made in the tree. This is my colleague Joe Rossetti's 10-year-old Cirsus canadensis, the red bud. And he actually, I don't know what happened beyond the cut here, but he could have actually fixed this by bolting it back together. This is a fairly clean break, but he still would have had a bad branch union here. And there are ways to prune these, and you want to keep an eye on these and should prune them when they're small. But if you let it get big enough, sometimes the only thing you can do is cable the two pieces together to take some of the load off of this, this union. Any questions about roots and branches?
So there was no question, but Bill Whitaker just wrote in Bartlett pear. I wasn't sure. Was that a question, Bill?
That was a comment.
Oh,
So
okay.
the reason, if you want to see bad branch unions, like this one, go and look at a Bradford pear. They have that really nice shape because they have all bad branch unions. And if you've ever seen what I call hand of God pruning, where basically the whole top of the tree explodes and falls off, and you're left with a stump with a lot of broken wood at the top, that's because all of the scaffold branches on a Bradford pear are attached to the trunk like that. So yeah, Bradford pear is a really great example of bad branch unions. That's why a lot of jurisdictions won't give developers credit for planting them, because they're just creating problems for the homeowner down the line. All right, now let's talk about tree growth and wounds. And in this, we're going to talk about the compartmentalization of decay in trees, also known as coda. So remember, only the meristem can make new cells. So trees can't replace lost tissues. What they do is they seal off decaying wounds and they create a box around that decay. Some of the barriers, some of the walls of that box are laid down during growth. Others are created in the heart when heartwood is created. And in the sapwood, some of those barriers are initiated after the wound, and the living parenchyma in the sapwood can reinforce some of those barriers because of those chemicals that they produce. So what does it look like? This is a diagram of the inside of the tree. These gray the red and the brown, these are growth rings. This would be the parenchymal rays that I pointed out. And this box that the tree builds around that decay consists of 6 walls like any box, but there are 4 different types of walls. Wall 1, which is the top and the bottom of the box, is the weakest wall. And what that is that clogged up vascular tissue that tries to keep the decay from moving up and down through the tree. In heartwood, it's laid down as part of creating the heartwood. In sapwood, it is created after the wounding by the parenchymal cells. Usually what's going on is they're pushing resins and sap and things into the vascular tissue in order to clog it up. Wall 2, which is the next weakest, is the wall at the back of the box. And this is that growing season barrier that I mentioned at the end of the growing season, and that's there to keep decay from moving further into the tree. Wall 3 is also laid down during growth, and it's those parenchymal rays, and that's the strongest of the walls that are created immediately after wounding, or that are present immediately after wounding. Those make up the sides of the box, and they're there to keep the decay from moving further around the trunk of the tree. Wall 4 happens after the wound, and that is the new growth that occurs after the tree is wounded. So if we look at a cross section, we can see wall 4. So the wound occurred. Wall 1 is this green stuff, which is clogging up the vascular tissue to keep the decay from moving up and down. Wall 2 are these growth rings that's trying to keep the decay from moving in deeper. And then wall 4 is, remember I mentioned you've got the callous tissue that's growing these rings. That's kind of where the cambium ends. And it is also laying down stronger new wood that is keeping the decay from moving into that new wood. So it's protecting the new wood from the decay here. If you look at it in a cross section, you can't really see wall 1, but wall 3 is here and here. This parenchymal rays, wall 2 is here. And then wall 4 is the one that is trying to seal that off. This picture is a little bit incorrect, unless you're talking about, say, decay that got in because somebody nailed something to this tree, a very small hole that did not create a need for these, we call these ram's horns, this wound wood to grow here to seal it off. Otherwise, you would still be able to see in some of these earlier growth layers, you'd still be able to see this rounded off wound wood. And on Saturday, I shared these with you to look at. This was a wound that occurred on a branch on my red maple. The tree was trying to close it. You can see it didn't do a very good job of controlling the decay that got into the wood that existed at the time of the wound. This is all new wood that grew after the wounding. The tree is doing a pretty good job of building its wall 4 and would eventually bring these two together, but it was doing a really bad job with walls 1 through 3 in that it was not preventing lateral movement or movement deeper into or along the branch. That's probably why it eventually got pruned off, is because all of this decay eventually killed the tree or the branch, or weakened it to the point that it got cut off. On a bigger scale, this is a tree down at Mason Neck State Park, and you can see some of that wound wood. So this is the wall 4 formation, but you can see that wound went way up into the tree, and that's the seam where it is starting to graft together, and that cambium is starting to constitute itself all the way around the tree. And over time, this seam will disappear, but the decay and the wound will stay there. The tree does not grow the new tissues to replace the lost tissue. One last thing I wanted to talk about before we get into pruning is tree growth and hormones. Remember I said that you've got primary meristem or apical meristem in the buds at the ends, at the tips of all the branches and twigs. And they produce a chemical called auxin, which works with other chemicals in the tree to suppress the growth of those lateral buds. So here you can see those lateral buds. They go all the way into the center of the tree because they're formed when it was a shoot, and they start and they keep growing a little bit to get out to the, stay out at the edge of the tree. If something happens to that lateral bud, a storm comes in and knocks the top out of the tree. In the case of the picture on the right, a bug comes in and kills the ends of all the branches. Those lateral buds can be released to create new shoots. Now, the function of those new shoots is to replace lost photosynthetic area. It is frequently a sign of disease, particularly in the disease or pests in the canopy of the tree, but it can also be an environmental response. If that tree was growing in the forest and you cut all the trees down around it, it might send out, release some of those lateral buds to send out new shoots. And with those buds, or with this suppression, you can get different types of trees. You can get an excurrent tree, which would be characterized by a pine, which has a very strong central leader and smaller branches on the side, or more of a decurrent tree, which would be like an oak, which will have no branches up to a certain part, and then it'll start growing those scaffold branches, particularly if it's grown out in the open. So before we get into pruning, are there any questions? And Nora, do you want to take a short break? Because I've been talking for over an hour.
So there's one quick question from Catherine. I think it's the tree that you were showing from Mason
Yes,
Park.
I see her question.
Okay.
It's not a lightning strike. Lightning strike might look something like that, but in this case, it could have been an old wound from a lawnmower or a string trimmer, which may cause the cambium for a long way above the wound to die. It could have been a soil pathogen that got in there because it looks like the whole butt of that tree is rotted out, and there's probably heart rot going all the way up into the tree.